Introduction to primary cell culture. Because they are derived directly from living tissue, primary cells maintain physiological relevance and thus find increasing use in life science research and pharmaceutical drug discovery. Compared to immortalized cell lines, however, primary cells can be finicky and present challenges during culture unless proper care is taken. Cell Applications recommends the user follow the steps outlined in these video segments to achieve optimal results with their primary cells. Receipt, storage, and transport of primary cells. Upon arrival, immediately remove the cryovials from their dry ice packaging and transfer them to a liquid nitrogen storage tank. During transport, always keep the cryovials submerged in dry ice. Do not at any point expose the cryovials to ambient temperature, as this could damage cryopreserved cells and compromise their performance. Preparation for primary cell culture. Prior to working with the cells, review the cell-specific steps and instructions provided by CAI. Adhere to standard, accepted guidelines for sterile technique, laboratory safety and equipment, such as appropriate eye and skin protection when moving the vials to and from liquid nitrogen storage. Work with the cells in a Class II biological safety cabinet and culture them in a humidified incubator set to 37 degrees Celsius and 5% CO2. Turn on the water bath. Set its temperature to 37 degrees Celsius at least 1 hour in advance, and check the temperature with a thermometer prior to thawing the cells. Ensure that you have the optimized cell-specific growth media. Also, confirm that your cell culture flasks are suitable for primary cell culture. We recommend using Corning, Greiner, and other appropriately treated flasks. To reduce the chances for contamination, use 70% isopropyl alcohol to wipe down working surfaces, media bottles, and thawed cryovials. Thawing primary cells. Fill an insulated container halfway with dry ice pellets. Retrieve the cryovial from liquid nitrogen storage and immediately submerge it in dry ice until ready for use. Turn the vial cap 1/4 to release any liquid nitrogen or pressure that might have built up in the cryovial, and then retighten it. Thaw the cells quickly by placing the lower half of the vial in the 37 degrees Celsius water bath, monitoring the vial closely as it thaws. Take the vial out of the water bath when there is still a small amount of ice remaining in the vial. This step takes approximately 60 to 90 seconds. Do not let the cells thaw completely. Decontaminate the vial exterior with 70% isopropyl alcohol. Plating of primary cells. Remove the vial cap carefully and do not touch the rim of the cap or the vial with your fingers. Resuspend the cells in the vial by gently pipetting 3 times with a 1 ml pipette. Be careful not to pipette too vigorously as to cause foaming. Pipette the 1 ml cell suspension from the vial into the T75 flask containing 15 ml of growth medium. Cap the flask and rock gently to evenly distribute the cells. Loosen the cap to allow gas exchange. And place the T75 flask in the incubator. For best results, do not disturb the culture for at least 4 hours. Change to fresh growth medium after 4 hours, or the next morning, to remove all traces of cryoprotectant. Change growth medium every other day until the cells reach 60% confluency, and double the growth medium volume when the culture is greater than 60% confluent, or for weekend feeding. Subculture of primary cells. Do not warm the trypsin EDTA solution or any other subculture reagents to 37 degrees Celsius. Also, be sure to use Cell Applications trypsin EDTA solution, which has the correct trypsin EDTA concentration. Subculture the cells when they reach 80% confluence. Aspirate away the medium from the T75 flask. Wash the adherent cells with HBSS and remove by aspiration. Pipette 5 ml of trypsin EDTA solution into the T75 flask and rock gently to ensure the solution covers all the cells. Aspirate 4.5 ml of the trypsin EDTA solution immediately. Recap the flask tightly and monitor the trypsinization process at room temperature under an inverted microscope. Watch the cells become rounded while they are still attached to the flask. This may be about 1 to 2 minutes after the trypsin EDTA was added. Do not over-trypsinize by waiting for the cells to detach. Instead, detach the rounded cells by hitting the side of the flask against your palm. Pipette 5 ml of trypsin neutralizing solution into the flask to inhibit further trypsin activity. Transfer the cell's suspension from the flask to a 50 ml sterile conical tube. Add 5 ml of trypsin neutralizing solution to the flask to collect any additional remaining cells and transfer them to the same conical tube. Examine the T75 flask under a microscope. If there are still more than 20% cells remaining in the flask, repeat the trypsinization steps and add the cells to the conical tube. Centrifuge the conical tube at 220 times G for 5 minutes to pellet the cells. Aspirate the supernatant from the tube without disturbing the cell pellet. Flick the tip of the conical tube with your finger to loosen the cell pellet. Resuspend the cells in 2 ml of growth medium by very gently pipetting the cells to break up the clumps. Count the cells with a hemocytometer or cell counter. Pipette 30 ml of growth medium into a T175 flask or flasks, depending on the cell number. Cells are seated at 5,000 cells per square centimeter. Inoculate the T175 flask or flasks. Cap the flask and rock gently to evenly distribute the cells. Loosen the cap to allow gas exchange and place the flasks in the incubator. And that's it. Following these guidelines will provide you with healthy cells and solid data. Learn more at www.cellapplications.com, where you will find protocols, references, and contact information for our technical team.